Hey there, ladies and gentlemen, boys and girls. Today we're going to talk about perhaps one of the most important topics that we're going to talk about all year because it really sets the groundwork for what we're going to be doing and talking about for the rest of the year, and that is life. All right, how does life work? How does it exist? And today we're going to talk about something called the central dogma of biology. This is a main theme for all of life and our understanding of how life goes, from the simple bacteria to the most complex big blue whale. All right, and every life that's ever existed on this planet, we believe, has followed basically this central dogma. Dogma is just kind of is a term that means kind of like a series of beliefs or like a series of ideas. All right, that you kind of follow. And pretty much every topic, even though we might not talk directly about it, every topic we're going to be talking about for pretty much the rest of the year revolves around this idea. And a lot of this first semester we'll spend talking about more specifics about each one. But right now we're talking kind of as a big picture conceptually. So this is the big one because this really sets the ground. All right. And so far we've been kind of building up to life. We've been talking about macromolecules, and now we're at the point where we can start talking about how those proteins, carbohydrates, lipids, and nucleic acids start to find that right combination, start working together to create that most basic form of life. And so when we think of this idea of first life on this planet, right, there's lots of different ideas on how to do this, but we think probably the first life on this planet was not some fish swimming in the sea or some mammal walking on the ground. Probably something almost unrecognizable as life, because it was probably unicellular, probably lived in the oceans, and it doesn't have a brain, it wasn't thinking, it wasn't sentient, it didn't have thoughts, right? It just barely met those criteria that we talked about to be considered living, all right? And so we're going to talk about how we could go from that very basic organism all the way to the vast type of life that we have on this planet. And it starts kind of this idea. So let's talk about this central dogma. The central dogma is this idea that organisms have DNA. Right? DNA can replicate itself. Right? DNA can replicate itself, make more DNA. The DNA is coded to make is a code to make RNA, which we'll talk about in a moment. And those make proteins, all right? So let's actually kind of work about this. Proteins, we mentioned before, these are the workers, all right? I like to use that analogy as them being workers, because any kind of function that the cell needs to do, all right, proteins are involved. Those functions could be from everything from carrying oxygen to different muscles in your body. Literally, the muscles in your cells that contract are due to specific proteins that are moving in and out, or that are contracting and releasing those cells. We talk about within the cell, the things, without those proteins, we couldn't do all kinds of functions. We couldn't relocate protein. We literally use proteins to make proteins, right? Then we have different proteins, send those proteins to different parts of the cell. We have certain proteins that are involved in turning our food into energy, or turning light energy into food, all right? Anything your cell needs to do, proteins are involved, and the way we make those proteins is from the instructions that we learn from DNA. Right? DNA is a nucleic acid, deoxyribonucleic acid. All right? A lot of people think it's like, well, it's kind of like the brain of the cell, and I don't really care for that terminology because it makes it sound like it's doing something, and it's not. All right? It's not doing something. It is an instructions. All right? It's the instructions on how to do it. You can think of it as like a Lego kit. Right? When you get in the box, open a box of Legos, and there's the instructions. You don't just lay the box out or the instructions out and then the Lego kit is built. No. It tells you what you need to do that. All right? And so I like to do this with like a little analogy as we start talking about kind of how this central dogma works, when we start talking about the different variations of complexity of life. All right, I like to do a little a little kind of analogy here. Because this is an analogy that I would hope that you can remember throughout the year, because every unit we talk about, we come back to DNA and how it relates to making proteins. All right, so let's think about it this way. All right, I have a little science here book. All right, This is a book. It's very, see how it's very small here. All right. This would be my DNA. All right. And what this is, and so I'm going to kind of go back and forth between talking about the book and DNA. All right. What this is an instruction manual on how to learn learn science. All right. It's not very big. There's not a lot of instructions, right? It's a pretty thin book here. In fact, it's got about 60 pages. All right. Our DNA is the instructions on how to do something, right? And this is the instructions on how to learn science. But our DNA is not just one big pile of things. It's made up of little sections, just like this book here is made up of little sections. We call these little sections, we call them pages, right? So this one here has about 60 pages, about 60 genes. Each one of those sections is called, in our DNA is called genes. So we may have a long strand of DNA, but each little section is a gene. And each one is the instructions for something else. Just like in here, each page has a little bit different information than a different other page. All right, so this is pretty simple. This is only about 60 pages here, right? So I can learn science from this, right? I could, do the basics of it, but I'm not going to be able to get very much from it. It's going to be very, very simple, my understanding of science from this, just because there's only so many pages, so many genes. Any organism that has just this thin of an instruction manual, this much DNA, is going to be a pretty simple organism. They're not going to be able to do a lot of different complex functions. Since they have very little DNA, they can do very few workers, and thus can only do a few things. Kind of put an analogy in an analogy here. If you think about workers in a factory, if you only have 5 workers, you can only do so much. If you have 10,000 workers, you could do a lot more. But right now, we only have about 60 genes. Very simple. This is probably what we think is kind of most of the first form of life was very, very simple. All right, but now let's say we're able to get more genes. All right, there's a whole lot of ways that we can do this, but let's say we get more genes here and we get a little bit of a thicker book. Right, this one's here is about 300, a little over 300 pages, about 350 pages. This here is a little bit more complex. I can learn a little bit more of it. More pages, that means more genes, DNA, which means I can make more proteins. In this case, I can learn. This organism is a little bit more complex. They can do more things. You can learn more from this than you can from this little tiny thing. All right? The more DNA you have, the more proteins you can have, the more genes you have, the more proteins you can have, the more complex things you can do, more workers. All right, so I can learn a little bit more from this one here, but it's not the real deal. All right, it's not the full thing. So if I was even to get even more, so this one here, it's still pretty thin. It's got the flimsy outside, but once I get to a certain amount of pages, a certain amount of genes, I can't have that flimsy outside to kind of hold it together anymore, because now I'm getting to over 1,000 genes here. I'm going to need something to kind of contain this, so I have a nice little exterior here. All right, in a cell, this would be the nucleus. All right, where we have something to contain all of our beautiful information here. I have over 1,000 genes here now. 1,000 genes means 1,000 genes. It means a lot of DNA, which means I have 1,000 proteins I can do. I can learn a lot more information from all of these pages than I can from the other ones. All right, so as we go from very simple to much more complex. Now, one of the benefits is when we get to this kind of level of complexity, right, where we're so complex that we literally have to have kind of a cover to kind of cover all of our information to keep it safe. Right? Once we get to this level, we can start creating proteins that do very, very, very specific jobs, like have an entire set of proteins that talk to other cells. And you can code them on the outside of the cell, put those proteins there that talk to other cells. And so all of a sudden, we go from one cell being able to talk to the cell next to it, and not just talk to it, but start to work together. Okay, maybe this one is spending time doing one function and this one's doing another function, and they're working together. They're almost collaborating. And now we start getting multicellular organisms. Organisms, because they're complex enough, they're able to work together. They become a much more complex. Now that they, not only are each one of these cells more complex than the original one because they have more proteins, more DNA, but now they are working together to do that. This is where we start to first see our our first multicellular organisms. But it took a long time to go from this very, very thin, short, small amount of DNA to a very complex form of life. All right. And so this is where we see now, and even today, this form of life here still exists today. This isn't just a precursor to previous forms of life. This form of life still exists today. All right. You see it all the time. Bacteria is like this. It has its benefits. All right. And so now we start kind of organizing our life into its most fundamental part, and it's a fundamental form of life, and that is the cell. Is that organism cells made up of the very simple, small amount of genes, small functions, typically unicellular because of that? Or are our cells made up of the complex, or the much more complex type of cell that can even become sometimes multicellular? All right, so this is our first way to start describing life. And the ones we're going to talk about is that very first simple one, ones that still exist today. And these are called prokaryotic cells. All right, these are prokaryotic cells. All right, so they typically, you can see different diagrams. Each one's a little bit different. I'm going to draw kind of a generic picture of a prokaryotic cell because there's a bunch of different life out there, and they all kind of look a little bit different. They have different types. All right, they are typically these very simple cells. Now, because they are very, very simple, you'll see a lot of times they'll have this membrane around them, very soft membrane, fluid, kind of like here, kind of this fluid membrane. All right, they'll have these little hairs that help them move. They're called cilia. Talk more about those structures. And like a tail-like function that almost helps it like swim. Some of them have these. This is called a flagella. We'll talk more about that. But these are all very, very, very simple organisms. Because they're very, very simple, we're talking, about 250 genes. And I say about because each organism is a little bit different, right? Each type of species. But I just want to give you a reference. Talking about 250 genes. We're talking about a thin book here, all right? And because of that, their eukaryotes are always going to be unicellular. These are only going to be made up of one cell because they don't have that level of amount of DNA to create those specialized proteins to become that communication, to become multicellular, right? They normally have, because they're very simple, they don't have as much DNA, right? Their DNA is normally just kind of floating around in there. Sometimes if it's a type of bacteria, it's normally circular. But the thing that defines a prokaryote, truly defines it, right, is prokaryote. Karyote means nucleus, right? A center place that holds DNA. This pro means before, so this is before the nucleus. We're still at that very simple stage, all right? So prokaryotes do not have a nucleus. Right? Instead, what they have is their DNA, that nucleus is what holds that DNA. In this case here, we have so much DNA here that we need to have this outer cover to kind of protect it. This would be the nucleus. They don't need that. Yeah. So they normally just have free-floating DNA. Right? Their DNA is just kind of floating around. One thing they also do have, though, is they typically have floating in that cell is like a jelly-like substance called the cytoplasm and cytosol. It's almost like Jello, kind of holds everything together. And then they have these little things called ribosomes. Ribosomes are proteins, very simple proteins, or they can actually be made up of RNA as well. They contain ribosomes. A ribosome's job is to basically help create proteins. There are proteins that make proteins. So to have any kind of life, we're going to need to do certain functions, and some of those functions are going to be done by proteins created by that limited source of DNA, right? And finally, we see a lot of things with prokaryotic cells because they're unicellular, because their DNA has very free-floating DNA, it's very small amounts of DNA, they typically can't have extra functions, specialized extra apps, or think of like apps on a phone or extra functions that you can do. These things called organelles. So they have no other organelles. You're probably familiar with some of these organelles from your past, like mitochondria, or chloroplast, or Golgi body, or endoplasmic reticulum. Hopefully you've heard of at least one of those. We'll talk more depth about those. To use all those things, they're great to have. If you have chloroplast and mitochondria, you can use light energy to create your own food, and then use your own food, that food you create using your mitochondria, very, very efficiently break down that food into energy. But to have all those things, chloroplast and mitochondria, you need to have, the foundation and the workers to be able to maintain those organelles. We just don't have those in a prokaryote. We just don't have those functions, all right? Because we are not complex enough. We don't have enough DNA or the right genes in DNA. I could say the correct genes. It's not always just about amount. It's which genes there are to create the right proteins to do those kinds of things. All right? So this is a prokaryotic cell, kind of what we consider a lot more of the basic units of life. It's a unicellular, free-floating DNA, no other organelles. They do have ribosomes, all right? But they're typically simple organisms that do that. Just because they're simple doesn't mean that they don't thrive, though. It's the last thing I want to talk about real quick. All right? Bacteria can spread everywhere. All right? We all get it. We'll all have to take antibiotics at some point. That's because bacteria, you have some sort of bacterial infection. If you eat food, your bacteria in your stomach is breaking down. Just because they're simple doesn't mean they can't be successful. They're just unlikely to be able to become more successful enough to become multicellular. We do not have any multicellular prokaryotic cells because they just don't have the DNA and the proteins to become that complex multicellular organism. Okay, I know that was kind of a long video. I apologize. It's just there was a lot I wanted to say there. So, just kind of go back, make sure you understand, think about those questions, some questions I posed to you guys in the agenda to think about different types of, about how that central dogma, and then a little bit about what it means to be a prokaryotic cell. We'll talk about the other more complex, these kinds of cells. We'll talk about these going forward. So, all right. Thank you guys very much. Good luck, and may the science be with you.